Okay, we resume our presentations and we go on with Indira Paudel from Nepal. Indira got her BSc in Agriculture from the Institute of Agriculture and Animal Sciences in Tribhuvan University of Nepal. Indira, please.
Salam, I am Indira Paudel from Nepal. Today I am going to present my research exercise seminar. The topic of my research exercise seminar is sap flow response to irrigation treatments and climate in nectarine tree. And this research exercise was conducted under the supervisions of researcher Dr. Saptahi Cohen from Agriculture Research Organization, Volcani Center. Nectarine, the scientific name of nectarine is Prunus persica, variety nectarina. It is originated from China and it is subtropical deciduous fruits. Although it is originated from the China, in Israel it is grown as rainless, hot summer grown fruit tree. So it is grown in the hot summer, so it requires irrigation to survive. We have general recommendation for the nectarine tree. It means how much water is required generally, but it is not sufficient for the Israeli conditions, and it is the main economical limiting factor for the yield and economic production of the fruit. And other factors such as soil or other factor of the specific orchard, just like the soil parameter, climatic parameter can determine the water use, and it is hard to predict, and it is not exact use of water if you use the general recommendation. So we have to develop the irrigation protocol according to specific orchard. To develop the irrigation protocol according to specific orchard, we have to understand the soil plant atmospheric continuum of water utilization. Soil plant atmospheric continuum of water utilization means how plant absorb water from the soil and how plant utilize water within the plant, and how atmosphere took the water as in the form of water vapor. And for this, the main driving force for to flow the water within the plant tree is stream water potential. And stream water potential, it means the potential energy required for the stream to draw the water and to drive the water within the plant. And the stream water potential has been shown to be the best indicator of water status in the various crops, including nectarine. Here, according to Noor et al. 2001 in nectarine tree, they found that the good relationship between relative yield in y-axis and midday stream water potential millipascal, and they found the both yield and fruit size was higher in the at high stream water potential. We are interested to know the stream water potential because although my topic is sap flow, it is because we want to irrigate. We would like to irrigate our orchard to maintain the given stream water potential. Sap flow means amount of water move within the xylem in the sapwood according to the transpiration demand of the climate, and this is the actual water use. Why we need to understand the sap flow? It means from the sap flow we can measure this can be measured with the sap flow sensor. It can help us to know how much water is needed to irrigate in the space in the particular orchard. It may also be closely related to stream water potential. It may also be a good parameter for our irrigation control, so that we can develop irrigation protocol for the fine and tune irrigation for the precision agriculture. According to different scientists, Smith and Allen 1996 and Orento et al. 2006. As I told, the sap flow method help to predict our water demand. Although there are various techniques as evapotranspiration, thermal plant heat balance, these techniques are based on evaporations, while the sap flow method is based on transpiration. So it is more specific than that of evapotranspiration and plant heat balance method. So we choose the sap flow method, and sap flow method can be measured by different type of sap flow sensor. These sensors are quantitative and it can be used to develop the automatic irrigation control program. Although it requires skilled manpower to installation, operation and analysis. There are a lot of sap flow sensors available in the commercial market, but we choose in our experiment thermal dissipation probes because it is simple, high degree of accuracy, reliable and economic. In this method, we are going to use two probes. It means the lower one is the reference probe, and upper one is the heated probes. The probes where heat will be supplied. Here the probes where heat will not be supplied, and this is the place where the water and mineral move in the place of the xylem of the perennial fruit trees. And this method was developed by Garnier in 1985 and it started to use in 1987. As I told, the sap flow in response to different irrigation treatments. The irrigation treatments that we are going to use is drying cycle. The drying cycle means we are going to restrict the amount of water in the certain stage of fruit development, so at that stage, so that it will not affect the yield and quality of the fruits. And it is effective to observe the response of sap flow to soil water deficiency and climate. Here are some graphs that justify that drying cycle can help to understand its response to stream water potential and sap flow. The x-axis indicates the stream water potential. The x-axis indicates the calendar day, while y-axis indicates the stream water potential in the form of negative. This is the stress tree. This is the non-stress tree, and we found that significant difference in midday stream water potential in the stress tree, but not in the non-stress trees. Similarly, transpiration rate, it means the sap flow. We found that the significant decrease in sap flow in the drying plot, it means in the stress plot, while we are not we are unable to found in the non-stress plot. It means drying cycle can be used as a to know the sap flow response. Without considering climate, we can develop the good irrigation protocol. So we have to understand the climatic phenomena, that climatic and canopy effect during the entire season of the growing period, because as you know, deciduous fruit, nectarine is a deciduous fruit, and in the beginning of the season there will not be the leaf, and when there is high fruit and leaf growth simultaneously. So canopy can affect on the water requirement of the plant. The evapotranspirated demand is affected by temperature, solar radiation, relative humidity, wind speed, and the evapotranspiration is the form of sap flow. So it can be affected by canopy size and stage of development of trees. This is justified from this graph from Togenti et al. 2004 in olive tree. Here daily sap flux gram cm square per day, and daily reference evapotranspiration mm per day. And they found that significant relationship between evapotranspiration and daily sap flux both in irrigated and non-irrigated. So climate can affect on the sap flow. from this graph we can say that climate can affect on the sap flow. So our objective is to monitor the sap flow from flowering to full canopy development, to determine the response of sap flow to stream water potential, soil water content, soil water tension in a drying and waiting period, to quantify the sap flow response to climate and plant parameter, especially vapor pressure deficiency, which is the function of relative humidity, midday stream water potential, stream contraction. Materials and method. The location of the experiment. Our experiment was located in the Khar Parut of Golan Heights in northern Israel. This is the place where our experiment is located, and this is just behind the Sea of Galilee. And the climate of experimental plot is semi-arid Mediterranean climate with rainless summer, and the rainfall in this area is nearly 600 mm per year, and it is from the October to April. So but our main experimental period is from April to September, when there was no rainfall. This is our experimental plot, which was 10 years old drip irrigated nectarine orchard. Experimental layout. We had altogether 17 experimental plots. Among them 16 which are denoted by this colored step, where we use the different type of irrigation sensor, and with 4 replication of each irrigation sensor, and in this plot we apply the same amount of water, while we have the different one plot with 6 trees, and it is different part of the orchard, and in this period we stop the water certain period of the time. And so it is special because we have just the two comparison, is a well irrigated and is a drying and waiting plot, and this all plot can be considered as a control plot with drying and waiting plot. And in the middle of the experiment plot there was the meteorological station, including anemometer, solarimeter, psychrometer and thermometer, while the each red, green, yellow, blue are the plot which was fixed by different irrigation center to control the irrigation level. And this X indicates the place where nectarine plant was nectarine plant, where the O indicates the plant where the sap flow sensor was located. And the experimental setup was done in the April, and the experimental plot was 15 meter square dimension of each plot, and the sap flow sensors, the irrigation sensors measure the data in each 13 minute average, while meteorological station measure the data in each 15 minutes from beginning of April to end of July. Sap flow sensor, we use the thermal dissipation probes which are developed by Garnier, and we use to collect the data. We develop the automatic collection of the data for measurement, and we develop the probes in our lab, and we install in the plot. This is the heated probes with the 2 conductor wire. This sense the temperature difference between the two places, and this is the constant heating wire to heat the sensor. This is the heated probes, while this is then we develop this and we install in the plot the about 10 cm above the soil level. This is the reference probes, while above 10 cm above the reference probes we install the probes with thermocouple. It means heated probes, and we connect these probes to the power board and power board to the central electric supply. Here the central electric supply is which was in the middle of the experiment plot, and to the data logger to collect the data. And we have altogether the SAP flow sensor. We installed 36 in control plots, while in the drying and waiting plot we had only 6 tree. In the drying and waiting plot we have also the irrigation sensors. In each 6 tree with 4 irrigation sensor to know the response of irrigation sensor during drying and waiting. Measurement and SAP flow sensor operation, we compare between the methods. The heater operation method was alternate every day between continuous heating, which was developed by Garnier 1985 and start to use in 1987, and discontinuous heating, which was developed by Dor and Rochechu in 2002. In the continuous heating, we supply the electricity continuously within 24 hours, and we measure the temperature. We measure the data in each 15 minutes average. While in discontinuous heating, we stop the our system in the 15 minutes, and we start to make on the system in the 15 minutes, and we measure the data at the end of the heating and end of the cooling. So these two are different type of the method, and this discontinuous heating is emerging technology, while continuous heating is widely used and mostly used, and the discontinuous method was developed, and do believe that it can preserve it can check the thermal gradient between the sensors, and it can be safe, it can be economic because it saves the electricity. As I told, we are going to stop 15 minutes the system. But this system is this type of measurement was done in our lab and in the Nectarine first case, and we did not have any calibration factor because we got the different type of result from that two method, and we try to find out the calibration factor for Nectarine and between the two method, because from the continuous method we got high daily average SAP flow, while from the discontinuous method we got less SAP flow, and we use this calibration factor to analyze the whole of the data. It means we found nearly 19% less SAP flow from the discontinuous method as compared to continuous heating. Result, steam water potential, as I told already, that is steam water potential, we want to maintain the constant steam water potential. The seasonal course of steam water potential of the experimental plot, steam water potential in terms of negative bar is the y-axis, and from the beginning of the season to the end of the July. This is the different plot. It means the plot where the irrigation sensor was located as a control plot. As I told, we have 4 control plot and 1 drying and waiting plot. Within the control plot, there was not significant difference between the steam water potential, but the significant difference pattern of steam water potential was observed in the drying and waiting plot. And we had 2 drying cycle. The first was from the 20th of May to 29th of May, and the second was from 14th of June to the 3rd of July. And in the drying and waiting plot, we found the nearly from 8 to the 9.5 bar of steam water potential when we stop the irrigation. After re-irrigation, it start to decline, but after drying, it start to maintain the same as control plot. Similarly, in the second drying, we found that up to nearly 13.4 bar of steam water potential at the peak of drying, and it start to decline with re-irrigation. So significant difference between the control and drying and waiting plot was observed, and highly variable during the drying and wetting in drying and wetting plot was observed. Sap flow response, seasonal sap flow response to irrigation and climate. Sap flow and potential evapotranspiration mm per day and the seasonal time from the April to the end of the July, and we found a significant increase in sap flow with the season, and it start to decrease the increasing rate from the beginning of July, both in control and drying plot. And also the potential evapotranspiration was also increased with season, and it start to maintain the balance. It means it start to decrease the increasing rate in the July. And we found in the beginning of the season, it means in April, we found nearly 1.8 liter per mm per day per tree in sap flow, while we found that nearly 2.6 mm per day from the plot potential evapotranspiration. While in the end of the season, we found nearly 4 mm per day per tree sap flow, while the potential evapotranspiration was 6. And we found the different response in the sap flow in the drying and wetting plot, especially during the drying season. The sap flow was decreased, and after re-irrigation it start to increase, and similarly in the second drying and wetting period. The significant seasonal response from the flowering to full canopy development development on nectarine and climate was observed, and we found the significant difference with p-value p-value 0.003, and the significant difference between the sap flow in the control and drying plot. And from the entire season, we found that nearly 11% less sap flow in the drying and wetting plot as compared to control. Canopy conductance response, monthly midday canopy conductance. The midday canopy conductance GC cm per second and month May, June, July in control and drying plot, we found that significant increase in sap flow canopy conductance both in control and drying plot with month, while the difference result was found in the May and June but not in July. It means in the May and June we have the drying cycle but not in July. So we found the canopy difference in canopy conductance result because of the drying period. No, it is not because of the different plots. Thus, the significant increase in canopy conductance from the flowering to full canopy formation was observed, and significant response in the control and drying plot in the May and June was observed, as I already told that it is because of drying and wetting period. And the sap flow response, the seasonal sap flow response, monthly, midday average climatic parameter with monthly, daily average potential evapotranspiration and sap flow with the radiation, watt per meter square, temperature, degree Celsius, relative humidity percentage, wind speed meter per second, potential evapotranspiration flow in the control and drying plot. We found that from April to May, June, July, every parameter is increasing, and the relative humidity is near about 50, around the 50 range, and the wind speed is also higher in the July, and the potential evapotranspiration also increased with the increasing the summer peak. And the sap flow was found different in the, we don't have sap flow result in the April because still we were installing the experiment in the drying plot, and here in the May and June we found the different result of the monthly average sap flow but not in July. And from this all analysis, we found that 68.68% of total evapotranspirative demand of the experimental plot was fulfilled by sap flow, and the difference in sap flow in drying and wetting plot at drying and wetting period. As I mentioned already, we found everything difference during because of drying and wetting period. So we try to find out what was going on during the drying and wetting period, and we try to find out the sap flow and canopy conductance during the drying and wetting period, the diurnal response of sap flow and canopy conductance at 24th of June, that means second days of drying peak. We found that sap flow in the control plot was near about 8 liter per hours, it means sap flow liter per hours and diurnal time, and while in the drying and wetting plot we found maximum of 6 liter per hours. While the canopy conductance was observed near about 7 cm per seconds from the control plot in the peak, and the drying and wetting plot we observed 4 cm per seconds. And the stream water potential in the drying plot we observed 14 bar, it means its decrease in stream water potential, while in the control plot we found the 9 bar. From this we observed that significant decrease in sap flow and canopy conductance and shape, and from the here we found that the sap flow is high in the was high in the middle, but the canopy conductance was high in the beginning. From this it is clear that says the stomata reaction make the strong effect on the sap flow. And the sap flow response to climate, we found the significant sap flow response in the mm per day per tree and PET potential mm per day. Significant relationship between the sap flow and potential evapotranspiration. Similarly, monthly diurnal sap flow course in the well irrigated period also found the significant increase with month, and monthly diurnal sap flow response to vapor pressure deficiency. We found that the vapor pressure deficient in the May we found that maximum is 0.8, while in the June we found that 1.5, and in the July we found that 3.5. The significant seasonal and diurnal response of vapor pressure deficient with sap flow was observed, and at the peak of VPD we found the sap flow was peak. It is because of gas, it is strongly affected by gas exchange phenomena of stomata. Average midday canopy conductance versus vapor pressure deficiency at the first and second drying period. At the canopy conductance and the vapor pressure deficiency, we found the significant relationship between the canopy conductance and vapor pressure deficiency, and we found that in the morning with the high canopy conductance, but in the afternoon found less canopy conductance, and this is because of the stomatal activity. So we found everything difference during the drying and wetting, and we try to understand what was going on during the drying period. This is the sap flow, this is the stream water potential, and this is the response of individual probes, and it is stream water potential. And we found that significant decrease with drying and increase with re-irrigation. And we try to normalize this factor with the try to find out the compare with the control plot, and here with a relative percentage decrease in sap flow during the second drying period to control plot, we found that 27% decrease in we found that 27% of sap flow has decreased as compared to control plot with excellent relationship between sap flow and percentage decrease in sap flow and the stream water potential. Similarly, the relationship between the sap flow, soil water tension, soil water contents during the drying and wetting plot was with a significant relationship was observed with the midday tension and midday sap flow, and similarly the midday soil water contents and midday sap flow. And similarly the relationship between the sap flow and stream shrinkage during second drying and wetting, we found the significant, this is the y-axis is the stream shrinkage, and the midday sap flow. We found the significant negative relationship between the sap flow and stream shrinkage. We found the significant negative relationship between the sap flow and stream shrinkage, and we observed the irrigation sensor response in the 3 irrigation sensor response during the drying and wetting, and the soil water tension, it means tensiometer responded earlier, it means significantly, and then soil water content, and then stream shrinkage. The summary and conclusion from this, we can conclude that the significant relationship found between the sap flow and potential evapotranspiration, vapor pressure deficient, and plant and soil water status, and significant difference between the well water and drying plot, especially the 11% seasonal and 27% at the second drying and wetting peak was observed. And Noor and Cohen in 2003, they found that in apple tree, they found that nearly 20% of transpiration was decreased and during the 20 days of drying period. Thus, the sap flow response to irrigation in climate was quantified and seems to be the one of the best tools for estimating water use according to seasonal development stage of nectarine and climate. Thanks to all participants, the Foundation Miracles at JNK to Canada, Mrs. Isabel Alder, USA, my advisor, researcher Dr. Saptahi Cohen, and my research groups, the Hebrew University of Jerusalem, Faculty of Agriculture, Dr. Amos Noor, Sasha Stulki, and Enigal from the Northern Research and Development Organization.
Thank you, Indira. Yes, Zerai. One at a time.
Yeah, one at a time.
You can ask more
Yes,
questions, but one and...
Okay. Thank you, Indira, for your presentation. You took sap flow measurement from stem.
Yes.
At what height?
What?
What height from the ground?
It's near about 10 centimeter above the ground.
Okay.
Trunk.
Was that the standard? I mean, yeah, the reading may be different, right?
Yes,
you keep on.
It's not. It normally done in the about the 10 centimeter above the soil. It means soil level in the trunk, in the main trunk. Okay. But I don't know it is standard or not.
You know why I'm asking that? Most of your R squares, the variability explained by the models you used, about 60%, 56%. So I even saw on some 56% of the variability explained by the model you used here, and it's significant.
Yes, this is the instrument. It for to sense the stream contraction and relaxation. It is not exactly the sap flow sensor.
No, whatever it is, but you are using this for some conclusion, right?
Yes, but it is installed near about 30 centimeter above the sea.
Yeah, look, the R square is 5.56.
No, it is. It means it is less responded with the sap. It means water deficient.
No. That tells you the variability explained by that line.
Yeah. I know. Strong
You know,
correlation.
if you want to predict even using this model, the predictability of R will be less than that one. You can't say anything about it. You are even 44% unsure.
So I recommended the last tools, the stream shrinkage is the last tools for the...
Even on the graphs above this one, there are also R squares.
It's already 94 and 88.
Go further up.
I don't think that there are near about... I think in the sap flow... In the sap flow and potential evapotranspiration, I think you got the 66%.
Yes.
But that is the different parameter. I'm comparing the sap flow and relationship between the sap flow and climatic parameter.
But if you got 66, that is poor at all.
No, it means 66% of the climatic demand is fulfilled by the sap flow. That is the m of the.
No. Okay, are you referring R square? The R squares
No,
tells you the variability that is explained by.
Here I got 94. I don't.
Here
Yeah,
you get
it's the
65.
Yeah, it means the climate totally. It means climatic demand totally can't be fulfilled by the sap flow.
No,
That
it's
is
not
my-
telling you that. It's telling you the variability explained by that formula, by that model.
Well, the question is right. The question is, what is the subflow supposed to represent? If it is supposed to represent the water taken up by the plant, then it's not necessarily has to be in a high correlation. It says only that the potential evapotranspiration, the daily potential evapotranspiration, is not in high correlation with the subflow. The real question that you raise is correct. However, you have to look at the correlation between the subflow and the daily water demand or water use actually by the plant. Is that right, Chaptai?
Yes, but I think in this case, we're talking about a method that was calibrated already. We have a calibration, and you should look at the standard errors in the graph, right? You have on some of the figures, let's say on the seasonal course, you have the error bars, which are standard error. And if you know standard error, plus or minus one standard error, that means 2 standard errors is the confidence interval for 95% for the mean. Okay, so you can see. And these are usually the means of, let's say, for the blue line here, towards the end, we were measuring 30 trees. Okay, that's maybe the standard error for measuring 30 trees, which you can see is a wide standard error for such a large sample, right? And that's why we measure so many trees, because it's highly variable from tree to tree. So you can see that even so, our confidence interval gives us, let's say we can, with this method, if we measure enough trees, then we can determine within, let's say, 10% what the real water use of the tree is. Are you following that?
We tell you the difference between means in that measurement. That I'm referring if she's using that model having 4 R square to infer something, I mean, I don't think that is fair.
Well, that's what we do when we look for relationships. We actually look for a relationship for something that's functionally similar, right? We have a measure of the dryness of the tree, which we use as stem water potential, or we look at the water content of the soil, which we have a real functional relationship between water use, or we should have. And then we look at that relationship. It looks more or less linear. We put a line in and we see how much of the variance is explained by that model. Okay, and this actually we do use for irrigation control if we have a good correlation that explains much of the variance, and then we get good results in the field. Whether it's 100% true, so we always have some variance due to some other factor. And models are approximations to what's going on in nature. Okay, so we dealt with, there's the statistics of the sample and how sure we are with that we have the right mean, especially when we're using a calibrated method. And then we have the question of a model to explain some of the functional relationships that we suspect are there in nature, which we want to use for irrigation control. Is that okay? Okay.
Indira, I have a question. Can you please go back to the basics, because I think I didn't understand how does the machine work? How does it measure the sap flow? Like,
So
I understood that you heat, like you're heating up the probes, but I don't understand how does it measure it with the heat? How?
it means in this we install the 2 probes One is just as a reference probe. It means in these probes we are not going to supply the electricity. It is just a reference probe. Here we supply it supply the electricity. It means with the thermocouple, which sends the temperature difference between the temperature difference, and here it sends the thermocouple sends the how much difference in the temperature in terms of velocity. It means from this
In terms of what?
in terms of velocity, the temperature difference is translated in terms of velocity, by
Indira,
sap flow velocity.
What happens to the heated sensor when the flow is rapid?
When the flow is rapid, it starts to sense the difference in temperature.
So it cools the heated sensor is heated at a constant rate. And when the sap
Flow
flow
is start to cool.
So the smaller is the difference between the two sensors, then it means that the subflow is larger.
Yes.
Okay.
So it's convection.
Larger.
Okay, thank you, Indira. One thing I want to see is the relation between potential evapotranspiration and water potential. Again, the water potential of the stem, I think this one is a large plant.
Which one? Which one? Which norm? Can you remember the slide
I
number?
saw in the graph the values of water potential, which is about 14, less than even 14, more of them.
This one?
Yeah, 14 bar.
Yeah.
It's the maximum is 13 or 14 bar.
The maximum is 13.4 bar.
But this is a large plant, and
It means
I don't-
we stop to irrigate normal. It means up to 14th of June we stop as the control plot equal amount of water. After that we stop 10 days of irrigation. We completely stop to irrigate 10 days. And the drying at the 10th day, it means 10th day, it means last day of drying peak, we got the 13.4 bar.
but I think this is the value for annual crops, for maybe for wheat or sorghum. I can say that 14 bar is very small in subflow. I don't know.
That
I'm
what
not
we
sure
got.
the physiology of the plant, but so we expose this plant to drought. At least we expect more than 2
No,
or
it
3.
is not exactly in the drought. You know, perennial plant have capacity to conserve the water. But
What
we just compare between the control that we try to maintain for the all plot the given water potential of 9 to 10 bar during the entire season, and we just try to find out the difference from those given stream water potential, and we found from 9 bar to it decreased to the it means in the negative it decreased to the minus 13.4 bar.
So minus 14, what did you use to measure this one? For example, you can determine the water potential of the stem using the leaf. So how do you measure the water potential of this stem? You
This
measure
is
the
the
water
automatic
potential.
measurement with the instrument. It means it is the automatic measurement, and that
Okay, I'll just say this is a very large experiment, as you saw in the you saw in the map of the experiment that it's a very large experiment, and Indira's project was to work on sap flow. So much of the data she didn't measure. Okay, she concentrated on measuring sap flow and looking into the relationships to other measurements. As we'll go, she'll maybe learn all the different techniques that we're using. But Northern R&D, the Northern Research and Development Company, had a team that was measuring water potential. Okay, and they were going out with a pressure bomb every few days, and Okay, and as for the technique to measure stem water potential, we cover leaves and we allow them to equilibrate with the stem for several hours, and then we pick the leaves and measure them in a pressure chamber. Okay? What? 1.4 megapascal is very small. You mean it relative? What value would you expect?
Maybe 3 or something like
Okay, you'd expect, perhaps you're familiar with olives or something. Some of our arid species will be 3 or 4 or 5 megapascals, but most of our agricultural, let's say, deciduous trees are in the range of 1 megapascal, of about 10 bars, when they're well irrigated. Okay, even citrus, when it's well irrigated, it'll be, let's say, 1.5. That's a more arid species. When I worked with citrus many years ago, so then it was common to have 20 bars or 2 megapascals. Today, when we irrigate much more, and water potentials go down. And don't forget that these are very high-yielding trees. The yield here is about 5 tons per dunam, which is about, Indira found a value of 1.4 for the worldwide average yield. So we're 3 times that. So we're well irrigated, optimal conditions, and a very good orchard that has low water potential. High water potential.
My name is Benny. I'm from the Department of Soil and Water Sciences. I have a question. You've showed a nice correlation between the stem flow and water content in the soil. And I wonder if you can use your data, you can extrapolate the data to other trees, other soils, or it's only resemble the situation in your experimental field.
Can I say again?
Can you show the correlation between the sap flow and water content in the soil, for instance? Do you expect to have similar correlation if those trees would have been grown in different soil? Or do you expect in the same soil same correlation with other trees, for instance?
No, if the soil water contents, it depends upon the soil parameter. It means how much the water holding capacity of the soil. If the soil have high water holding capacity, it will be different. It means it will be affected by the soil. It means plot, and it will be affected by the plot. It means soil.
So the soil is a factor?
The soil can be the factor because different place, it means different place of the orchard have different water holding capacity. The soil water contents, it depends upon the water holding capacity of the soil.
Okay, I suggest that we move on to the next presentation. Thank you very much, Indira.